Good morning or afternoon for many of you, and welcome to XenoTech's webinar, Navigating the Transporter Wave of 2013, a review of the 7 recent ITC publications on drug transporters. My name is Matt Beck, and I'm the marketing manager here at XenoTech. Today I am pleased to be joined by members of the XenoTech Drug Transport Group. Before we get started with today's presentation, I would like to go over a few housekeeping items. Here is an example of your go-to webinar user console. Using the orange arrow will allow you to minimize and maximize the user console. Please note that your audio is muted. If you have questions during the presentations, please post your questions in the questions pane seen highlighted here. We will have a Q&A session immediately following today's presentation to address questions posted via the questions pane. A copy of the slides and recorded session will be available in a few days after the presentation. We will send both links via email for your reference. These links will also be available via the XenoTech website in the coming days. Right now I'd like to launch a quick poll to see where our audience stands as regards to reading these papers. So I'm going to launch this poll, and it reads, have you read the 7 ITC publications? So go ahead and just respond to this so we have an idea of who's read these and who maybe needs a little bit more brush up. I'll give you guys a few moments to take this poll, and then I'll share the results with you. All right, I'm going to close the poll, and I'll share it with you. Looks like about 48% of you have read 1 or 2, but not all of the publications. 36% have not read any of them, and about 15% of you have skimmed through them. So no one's actually read through them thoroughly, so that's good. That's what we've done, and we're hopefully going to present some information to you so that you do not have to do that. So with that, I am pleased to have 3 members of XenoTech's Drug Transport Group with me today. I'm joined by Principal Scientist Greg Loewen, Senior Scientist Andrea Wolff, and Research Scientist Dr. Amanda Hayes. Without further ado, I'm going to turn it over to Greg, and he's going to get us started with a brief overview of XenoTech's Drug Transport Group. All right, Greg, I'm changing you to presenter now.
Okay, thanks, Matt. Thanks everybody for joining us today for this webinar. I'm going to start off with a brief introduction of XenoTech. So XenoTech is located in Lenexa, Kansas. We're a GLP compliant CRO founded in 1994 by Dr. Andrew Parkinson. We have over 100 employees. We have the global distributors throughout the world. We have partners including Sekisui, which is our parent company located in Japan, and then Cypex and Xenometrics and a few other ones for different collaborations. We were purchased by Sekisui Medical in July of 2008, and we are a part of the Next IND, which is a partnership with small CROs. The main focus of XenoTech is in vitro products and services to predict clinical drug-drug interactions as outlined in the FDA and EMA guidances on drug-drug interactions. XenoTech has 4 main service areas corresponding to the FDA and EMA's areas of interest for prediction of clinical drug-drug interactions with in vitro assays. They include the drug metabolism department, focusing on reaction phenotype and assays and species differences, as well as metabolite identification. The enzyme inhibition group, that they work on direct, time-dependent, metabolism-dependent inhibition of CYP enzymes, UGTs, and other enzymes. The enzyme induction group, focusing on the upregulation of enzymes in primary human hepatocytes. And then our group, the drug transporter department, focusing on transporters identified by the ITC group as being likely to be involved in clinically relevant drug-drug interactions. We do have some other discovery or late preclinical support studies, screening studies, if you will, and then all groups are supported by our bioanalytical group. We also have an independent consulting department mainly focused on these same areas, and they help investigate a particular compound in more detail, design in vitro and in vivo experiments for that particular compound to investigate any effects that have been seen either in clinical studies or preclinical species. They also write position papers when a compound is being submitted to the FDA. One of the things that makes XenoTech strong is our shared expertise across groups, and that's from our support groups. These groups help keep us running efficiently so the scientific core can focus on the science of your compounds. I highlighted some of the major groups, including our quality assurance group. They keep our facility GLP compliant. The knowledge management group, they write our reports and keep everything formatted up to the client's standards or to the ECTD document requirements. And then our analytical sciences department. I already mentioned these guys, but they take care of all of our analytical work. We can just pass our samples off to them, and they do a great job of getting the analysis done and done correctly. The drug transporter group at the XenoTech, we focus on the transporters requested by the FDA and EMA guidance, and we can help you with the study design for those. We use MDCK2, LLCPK1, and KCO2 cells for our transwell assays for PGP and BCRP. We use vesicles for E-Flux transporters such as MRPs, ESEP, PGP, and BCRP when appropriate. We use transfected cells, HEC293 and S2 for SLC or uptake transporters, and oocytes. We do have the capacity or capability to do oocyte studies as well. We do hepatocyte uptake or lysosomal tracking studies, and sometimes we partner with our parent company on certain assays and study designs when needed. The full list of transporters is available either on our website or it's listed here. And a lot of these transporters, there's multiple transporters listed on one line. So like all the OATPs are listed on one line. And then here's a picture of the drug transporter department at XenoTech. I'm the one on the right squinting here, and then Andrea and Amanda here who are also presenters. A big thanks goes out to the ITC group for publishing these papers. 7 papers from the ITC are included in the July 2013 clinical issue of the Clinical Pharmacology and Therapeutics. The title and lead author are listed here along with today's presenters and in the order of presentation. So these papers raise many questions and in some cases present similar topics from different angles. However, they do present the most current information and our best understanding of the topic today. They will ultimately advance the field. In today's presentation, we can only scratch the surface, but we will provide highlights from each paper, and you will come away with an understanding of what is contained in each paper. So before we turn this over to Amanda for the first presentation, we'll have Matt launch our second poll.
Yeah, thanks, Greg. I'm going to go ahead and launch this. We just want to know what's your main interest in transporters. Is it educational? You're just trying to learn more about transporters. You work in a separate phase. Mechanistic, trying to explain or predict in vivo ADME observations. DDI, predicting all potential DDIs, or submission. Are you guys looking to check the box before you're submitting to your NDA? Give you a few seconds to put your vote in. All right, I'm going to go ahead and close this, and then I'll share the results with you guys. It looks like, as you can see, about 48% are looking at DDI, predicting all potential DDIs. 29% are interested in the mechanistic aspect, trying to explain or predict in vivo ADME observations, followed by the educational. So, all right, I'm going to go ahead and hide this, and then we'll move forward with Dr. Amanda Hayes.
All right, thanks, Matt. So I'm going to start off by talking about the first paper the ITC came out with that was issued to address clinically important transporter polymorphisms in drug development. So the focus of this paper was on 2 transporters, OATP1B1 and BCRP, for the reason that both transporters have polymorphisms that have shown consistent clinical associations with multiple substrates. OATP1B1 is an uptake transporter encoded by the SLC-O1B1 gene. It's expressed at the basolateral membrane of human hepatocytes and functions in the hepatic uptake of numerous therapeutic drugs. There have been shown to be greater than 40 polymorphisms associated with this SLC-O1B1 gene. One of the most important ones, or ones that have shown clinical data, is this particular polymorphism that has a valine substituted by an alanine at position 174. And this particular polymorphism has impaired transport of statins, which has led to, or shows an increased statin plasma exposure. BCRP is an E-Flux transporter. It's a member of the ABC family, encoded by the ABCG2 gene. BCRP is widely expressed in tissues such as intestine, liver, kidney, and the blood-brain barrier. And it functions in the cellular efflux of a wide range of xenobiotics and endogenous compounds. In this particular BCRP polymorphism is also linked to increased plasma exposure of several different BCRP substrates. This slide is an example of a BCRP polymorphism in a clinical study that was done with statins. Rosuvastatin is a substrate of BCRP, thus linking BCRP to having a clinically relevant effect on the pharmacokinetics of rosuvastatin. Shown here is an AUC, or a concentration of rosuvastatin, that's twofold higher in individuals with this BCRP variant here compared to individuals with other genotypes who do not have impaired BCRP activity. And since clinical studies of this nature have been shown, a result of these has led to recommendations for genotype-informed dosing of statins. The authors also describe many other instances demonstrating the effect of transporter polymorphisms on the PK of drugs, and therefore, as a conclusion of this manuscript, they highlight a table of how to conduct future drug transporter association studies with respect to these polymorphisms. So as a first step, they recommend preclinical studies to be determined for a drug if it's a substrate of key transporters. This is normally done in vitro methods. If the transporter is a substrate or the transporter is known to play an important role in the disposition or response to the drug, pharmacogenomic studies are considered. But before that, DDI studies can be done with a specific inhibitor of these transporters. If a DDI produces a large effect on drug concentrations, or if the transporter is known to play a role in the disposition, pharmacogenomic studies can be considered. Interpretation of a large effect depends on the drug and its therapeutic window. So for example, if a therapeutic window is greater than 10 and there's a twofold increase in AUC, it is not considered important. By contrast, if the therapeutic window is small or around 2 with an AUC change that's greater than twofold, this would be considered significant. So in this table, they also outlined these pharmacogenomic studies where DNA samples are collected from subjects enrolled in these studies. They emphasize that sufficient numbers of subjects with variant alleles from different racial and ethnic groups should be included in these types of studies. And their overall conclusion is to apply a multi-scale systems pharmacology approach that incorporates all the data obtained from these in vitro and in vivo human studies. So this brings me to the second manuscript. I will discuss the emerging transporters of clinical importance. So in March 2010, the FDA released a white paper that provided direction and decision trees for key transporters to focus on when conducting in vitro and in vivo DDI evaluations. In that manuscript, they identified 7 main transporters that have compelling clinical evidence of involvement in clinical DDIs, and these include PGP, BCRP, OATP1B1 and 1B3, OCT2, and OAT1 and OAT3. The goal of this paper is to discuss emerging transporters of clinical importance and recommendations on how to study these transporters, and these include the MATE transporters, MRPs, and VSEP. So briefly to go through these emerging transporters, the MATE family, or multi-drug and toxin extrusion transporters, are comprised of MATE1, MATE2, and the splice variant MATE2K. All 3 isoforms are expressed abundantly at the apical membrane of proximal tubules in the kidney. and play roles in the secretion of cations and zwitterions into urine. They function as cation proton antiporters, and given their overlapping substrate specificity with OCTs, or organic cations, they have been thought to work in tandem with these specific transporters. So you can see here OCT2 is expressed at the basolateral membrane of these proximal tubules. And it's important to note that the antidiabetic drug metformin is probably the most famous example of a drug that is dependent on both transporters for clearance. Due to little information on the differences in the substrate or inhibitor specificity of MATE2 and MATE2K, oftentimes MATE2K is generally used as the model for both isoforms. Additionally, polymorphisms of these mates have been shown to have clinical effects in metformin-treated subjects. And here the ITC suggests using in vitro models for the study of whether a new molecular entity is a substrate or inhibitor of these transporters, which are often done in cell-based systems. Now moving on to multi-drug resistance proteins. They're also considered emerging transporters implicated in clinical DDIs, specifically MRP2, MRP3, and MRP4. These transporters are ATP-dependent E-Flux transporters, and they're expressed in many different tissues from hepatocytes, intestinal epithelia, and renal tubules. Accordingly, they're essential for the hepatobiliary and renal excretion of many anionic substrates, as well as some conjugates. In hepatocytes, MRP2 here is expressed at the canalicular membrane of hepatocytes and are important for the efflux of conjugates into bile, while MRP3 and MRP4 are expressed at the basolateral membrane and are important in the excretion of glucuronides and glutathione conjugates into the sinusoidal blood. Interesting note here, a complete loss of functionally active MRP2 at the canalicular membrane of hepatocytes has been associated with as the molecular basis of Dubin-Johnson syndrome, which contributes to conjugated hyperbilirubinemia. And a common test to study MRP function includes inside-out membrane vesicles. The bile salt export pump has also gained importance as an emerging transporter in clinical significance. BSEP is localized to the canalicular membrane of human hepatocytes and functions as the rate-limiting step for bile salt transport across hepatocytes for which there is no backup system. Thus, inhibition of BSEP can lead to cholestasis, as evidenced by BSEP polymorphisms that have been shown to cause progressive cholestatic liver disease. Although BSEP is unlikely involved in drug disposition, its inhibition may contribute to adverse cytotoxic events and is important to consider, especially if a drug exhibits hepatotoxicity during in vivo studies. And here, studies with isolated membrane vesicles are the best technical approach to currently investigate transport mediated by BSEP. Now, the transporters mentioned so far are all examples of transporters that can interact with a broad range of different drugs. However, it's important to keep in mind that for certain classes of drugs, other transport mechanisms may also be important. Two such examples are the human equilibrative nucleoside transporter, or ENT1, which is important in the delivery of certain drugs like nucleoside analogs to tumors, and the human peptide transporter, PepT1, which is important for the oral delivery of many peptide-like drugs. The model test systems for these transporters include anything from oocytes to cell lines and even knockout mice. So the summary for this manuscript are threefold: that new molecular entities that are actively renally secreted are recommended for in vitro investigation as mate substrates, in addition to investigation as substrates for the renal transporters OAT1, OAT3, and OCT2. Also, If signs of cholestasis are seen, retrospective analysis of inhibition of MRPs and BSEP can also help determine a mechanism of toxicity. And transporters like ENT1 and PEPT1 are examples of transporters that we don't clinically, routinely screen that can contribute to efficacy and distribution as well. So in the third ITC paper, I will be discussing transporters and drug development and follow-ups on some of the decision trees from the ITC. So in this manuscript, the ITC revisits the drug transporter strategy. The authors mention that these decision trees are not definitive and encourage flexibility in the timing of and selection of transporters to be studied. Briefly, transporter studies can be conducted in the drug discovery phase here, where they take into account the therapeutic area, co-medications, and other things like physiochemical properties of drugs. The second phase, where transporters can be studied, are generally done to build an understanding on transporter roles in drug disposition and their interaction profile. And the third phase includes mechanistic and investigational studies that are conducted for drug labeling and post-marketing commitments. Overall, it's important to keep in mind the central tenet is the clinical plan, which considers the therapeutic area, co-medications, and the patient population when designing these transporter strategies. The authors also present examples of decision trees. All new molecular entities should be evaluated as substrates for the E-Flux transporters PGP and BCRP in vitro. If the new molecular entity is hepatically excreted as a major route of elimination or greater than 25%, then the drug should be evaluated as a substrate for the hepatic transporters OATP 1B1 and 1B3. Similarly, if the compound is renally actively secreted, then evaluation of this drug as a substrate for the renal transporters OAT1, OAT3, OCT2, and MATES is also recommended. So this manuscript also highlights decision trees for each transporter subset. I'm not going to go into detail, but this is an example of one. I should mention that in addition to evaluation of these investigational drugs as substrates, regulatory agencies also recommend evaluation of these drugs' potential for inhibition of these transporters and include recommendations and cutoffs for these decision trees. And specific guidelines are highlighted in this manuscript. But to summarize the important points of this paper, the ITC here presented a flowchart for the timing of transport studies. They also discuss the integration of transport studies into the clinical plan, taking into consideration therapeutic areas, physicochemical properties, and PK of the drug. They also present 3 decision trees with respect to the E-Flux transporters, hepatic and renal transporters. Also, some of the highlights of this paper include a table that summarizes the approved compounds that failed the FDA's cutoff for clinical DDI studies, as well as a summary of the drugs that have post-marketing requirements or post-marketing commitments on those drug labels. And they also presented 3 case studies that discuss a few challenges in transporter DDIs with respect to applying PGP, BCRP inhibition decision trees, as well as complexity of OATP DDIs, and assessment of transporter-mediated DDIs for liver-targeted compounds. This concludes the portion of my ITC papers. I'm going to go ahead and turn it over to Greg to follow up with additional papers.
Thanks, Amanda. The next paper that we're going to talk about is the in vitro methods paper, published by first author being Kim Brower. So the first assay that we're going to talk about are vesicle assays. So in a cellular system, an E-Flux transporter pumps from inside the cell to outside the cell using ATP as an energy source. These cells can be ground up, essentially, and ground up and homogenized and turned into isolated membrane vesicles. A certain amount of the membrane vesicles are in the inside-out orientation, and in this case, the compound will be pumped into the vesicle by the E-Flux transporter. The right side out vesicles do not really have any function in these assays. These vesicles can be made from either SF9 or SF21 insect cells infected with a cDNA containing baculovirus, cDNA transfected mammalian cells such as HeLa, HEC293, etc., or from tissues such as human kidney and liver tissue. The homogenization portion is very important to be consistent between batches of vesicles and within the batch of vesicle itself. Substrates and inhibitors are listed within the article, and the most common transporters are MRP2 and BSEP, but this can also be done with certain MRPs, PGP, and BCRP. So the way the assay works is radioactive or fluorescent compound is the typical probe substrate, but it's incubated with a vesicle. It's taken up into the vesicle by the E-Flux transport in the presence of ATP, but not in the presence of AMP, because ATP is needed as an active energy source. At the end of the incubation, the vesicles are filtered out, then washed, and then that filter is analyzed for the content of compound that was trapped in the vesicles, either by liquid scintillation counting or LCMS. This can also be done through gel centrifugation through a gel matrix, and that's really good for compounds that have high protein binding or high binding to either the vesicle itself or to the plate. So a couple things that should be considered is the lack of trapping in the vesicle due to nonspecific binding either to the vesicle or the plate. You may need cofactors, especially for MRP2, requires glutathione. Cytotoxicity does not affect the vesicles as much as it would a cellulose system, so you can probably go to higher concentrations with a cytotoxic drug compared to a cellulose system. You can do things like explore pH gradients. They're less affected by that. And then the same system can be used over and over again, so you can make a big batch of these vesicles and store it frozen. And vesicles are available commercially. Next one is recombinant cell lines. Recombinant cell lines are cells that are transfected with full-length cDNAs encoding a transport of interest. There's numerous different ways to stably transfect the cells. They're kind of listed here. And then it's also possible to transiently transfect the cells, but that is not preferred. Most cell lines or most transporters are stably transfected now. Uptake transporters are expressed in HEC293, Chinese hamster ovary, and DCK2, S2 cells. And I'll talk about the experimental design of those in a few minutes. Oocytes are another option, but that's a transient transfection, so it's not used quite as much anymore. E-Flux transporters are typically transfected to LCPK1 or MDCK2 cells, and then the assay system is a transwell assay. So the SLC transporters, the test systems are transporter transfected cells grown in a cell culture plate. This can be done in an inhibition mode where the uptake of a probe substrate is measured in the presence of the test article, or the substrate mode where you measure the accumulation of the test compound in the cells. Typical cells or typical transporters are listed here. Some hepatic uptake transporters and renal uptake transporters. So this is a depiction of the cells grown on the bottom of a cell culture plate in the presence of the test compound. During the incubation, the test compound is taken up into the transporter expressing cells, but not the control cells. At the end of the incubation, the excess media is washed off, the cells are lysed, and the amount of compound in the cell is quantified. Typical inhibition results, so that's uptake in the transporter expressing cells compared to uptake in the control cells. As the concentration of an inhibitor is increased, the amount of uptake decreases. A couple comments on both vesicle and uptake transport assays. Accumulation in these test systems is usually very rapid. The kinetic should be studied in a linear range of time and protein concentration. So it's usually between like 1 and 5 minutes. Solubility and nonspecific binding should be considered. So solubility of these compounds, typically test articles can be added in up to 1% of a solvent such as DMSO. And if the compound is highly protein bound or highly bound to the walls of the plate or to the vesicles themselves or the cells themselves, BSA can be added, but you should consider the free concentration in the incubation and not the total concentration if BSA is present. There's no consensus on the best way to calculate inhibition constants for transporters yet, no best way to calculate a Ki, but there's a couple comments in here. So for competitive inhibitors, the IC50 value depends on the substrate concentration, whereas the Ki does not. The IC50 value will approach the Ki if the substrate concentration is far below the Km. And then if the Ching-Prosov equation is used to estimate the Ki, the probe substrate concentration should be within twofold of the Km. But this Chang-Krusov equation makes a lot of assumptions that may not be correct: single binding site, competitive inhibition, etc. So the best way to do a KI experiment is still being discussed, and we've done a few things at XenoTech as well. Moving on to bidirectional transport or transwell assays. Transwell assay is a polarized layer of cells grown up on a permeable membrane that separates two chambers. So the apical chamber is on the top side of the cells and the basolateral on the bottom. This is typically done for PGP and BCRP. Cell systems include MDCK2, LLCPKM1, and KCO2. Then the compound is loaded either to the apical side or the basolateral side. And permeability across the monolayer is measured, and the E-Flux ratio is determined. The E-Flux ratio is the B to A permeability divided by the A to B. Then E-Flux transporters typically pump in the B to A direction, and so an E-Flux ratio of greater than 2 indicates that the compound is interacting with this E-Flux transporter. It can be difficult to correlate an E-Flux ratio to extent of active transport in vivo, so even if you have an E-Flux ratio of 50, it may not indicate that there's some clinically relevant interaction. Some problems with this assay: the binding and metabolism can complicate a data interpretation, so recovery should be higher than 70%. Highly permeable compounds can saturate the transporter and give a false negative. Low permeable compounds may need an uptake transporter to get into the cell across the basolateral membrane to interact with the E-Flux transporter on the apical membrane. KCO2 cells can be used as a model for oral absorption, and these cells can be used for inhibition, but there's a few complications with that, several IC50 calculations, and it's in a separate publication by Benson and Carolyn Lee. Moving on to hepatocytes, there's a couple different ways to use hepatocytes for transporter assays. The first one is the oil spin method, and in the oil spin method, there's a long, skinny tube, long, skinny Eppendorf type tube with a layer of sodium hydroxide on the bottom, with some silicone oil over the top of that, and then they do the incubation with hepatocytes and the test compound. Test compound gets taken up into the hepatocytes at the end of the incubation, which is very short, maybe 30 seconds to 5 minutes. The tube is centrifuged, the hepatocytes pass through the oil, but the substrate solution stays on top. Tube is frozen and then cut at the oil layer, and now you can separate out the compound that's taken up into the hepatocytes from the rest of the media. The other way is the loss of substrate, so the incubation is done with the hepatocytes and the test compound. Compound is taken up into the hepatocytes. The hepatocytes are centrifuged at a slow speed to get them pelleted at the bottom of the tube, and then you can sample the supernatant and measure for loss of substrate. So anything that's lost from the substrate is either taken up into the hepatocyte or metabolized. The most elegant method is probably the sandwich culture hepatocytes. In this one, hepatocytes are plated on or put on plates. They grow confluent, cuboidal, form tight junctions between and bile caniculus between the hepatocytes, and then there's a matrigel overlay over the hepatocytes, and that's why they're called sandwich culture. The way this works is incubations are done with these cells in the presence and absence of calcium. In the presence of calcium, the bile caniculus between the individual hepatocytes stays intact, but in the absence of calcium, that bile caniculus opens up. So you measure accumulation in the cells with and without calcium. So accumulation with calcium is the total accumulation in the cell plus the bile caniculus. Absence of calcium, it's only the accumulation in the cell. So the difference is how much compound essentially was pumped into the bile caniculus. And so you can calculate a bile efflux quotient from that. The best way to summarize this paper is to summarize the tables. They give the best information in here. Table 1 goes over the vesicles, substrates, and inhibitors. Table 2 talks about the cell systems, substrates, and inhibitors, so the uptake and the efflux transporters expressed in cell systems. Table 3 lists commonly used equations for calculations of kinetic parameters in vesicles, cell lines, and hepatocytes. In a few minutes, Andrea is going to go over a much more complicated paper covering a lot more complicated equations. Table 4 talks about the applications, strengths, and limitations of the various in vitro transport assays. And then Table 5 is a really good table because it talks about the particular scientific questions that may arise during drug development. It talks about observations to support the in vitro transport assays, the in vitro tools to address each scientific hypotheses, the outcome of the in vitro experiments, and the possible follow-up studies. So it's a really nice table. Really tells you how to use these in vitro tools. So moving on to the next paper. This is the intercellular drug concentrations and transporters. So the most important concentration when considering a drug transporter interaction is the intercellular concentration, or at the site of expression. And for hepatocytes, BSEP, the most important concentration is the free intercellular concentration. But it's not easy to directly measure that. Some of the factors that influence the intercellular drug concentration and disposition in the liver: one is the blood flow and the mixing of portal and venous blood. And the overall goal of these assays, the overall goal of this is try to come up with a KPUU for the liver. That's defined as the steady state liver to sinusoidal blood partition coefficient from a bound drug. Because if you know that ratio, you can measure the Cmax of the compound, and then you can calculate the free concentration in the hepatocytes. So there's many factors that influence the free concentration in the hepatocytes. So one is the plasma protein binding in the plasma. Number two is the passive membrane permeability. Three is the active uptake from basolateral uptake transporters to concentrate compounds in the hepatocytes. Four is intercellular protein binding. Five is membrane partitioning, so do compounds stick in the membrane of the hepatocyte. Six is organelle sequestration, such as lysosomal trapping. Seven is basolateral efflux transporters, things like MRP2 and MRP3, 4, and 6. 7 is metabolism. 8 is metabolism, and then 9 is efflux out through the bile. So all these things will affect the total concentration in the hepatocytes. All these are affected. All these different things can be described by these different clearance values, and then the clearance values. Well, the clearance values can be used to make some generalizations. So a high permeable compound, a compound that can cross in and out of the liver freely, is going to have a KPUU of about 1. If a compound is highly metabolized or highly effluxed, either back into the blood or into the bile, it's going to have a KPUU less than 1. Or if there's active uptake or organelle or membrane distribution, the KPUU will be much, much greater than 1. And then this last panel in this figure represents all the transporters that may be involved in hepatocyte concentration. So the whole goal is to kind of come up with a PK model. And here's an example of one. So it's two compounds. The top set of graphs is a compound cleared by metabolism, and this one is cleared by active uptake into hepatocytes. So on these we have solid line is normal activity, dashed line is half activity, and then the dash and dot line is 2 times activity. So when the metabolic activity drops, the systemic concentration is going to increase, and the unbound liver concentration is going to increase as well. KPUU will increase. However, when uptake transporters are affected, now the Cmax, or the plasma concentration, is also going to increase. However, the concentration in the liver is going to decrease because you don't have as much being taken up, and it's also going to affect the KPUU as well. So that KPUU is going to drop. So the opposite effect between metabolism and active uptake. Again, on this paper, we can summarize a lot of the important points by summarizing the tables. So Table 1 gives examples of studies in which membrane transporters have been shown to alter hepatic drug concentrations, and those transporters are listed here. Table 2 talks about the in vitro, in situ, and in vivo models to estimate intercellular concentrations. Talks about the systems, the advantages, disadvantages, and examples of some of the parameters estimated from those assays. That includes vesicles, recombinant proteins, hepatocytes, sandwich culture hepatocytes, perfused liver, animal models, knockout animals, human in vivo studies. So there's a lot of good information in that table. Table 3 lists the direct and indirect methodologies for estimation of intercellular drug concentration. The table includes the direct or indirect method, the analyte or matrix analyzed, the detection method, and then the utility limitations and assumptions. I'm not going to go through all of these. It's pretty complex, summarized very well in the paper. And Table 4 goes over essentially some calculations used to estimate the intercellular fraction of unbound drugs. So you can use some of the data from these first Tables 2 and 3. We will go into some of these methods. So, but they can use things like physiochemical properties, monolayer permeability, hepatocyte uptake and binding, transport kinetics, all to come up with a PK model. So in summary, the current challenges: our understanding of intercellular drug disposition is rudimentary. So, for example, the trapping within the cell, we really don't have a good handle on what's going on there. Currently available in vitro and in vivo models have limited capacity or capability to quantitatively predict the impact of transporters on the intercellular drug concentrations. So we know there's an interaction, we just can't tell what it's going to do to the overall intercellular drug concentration. Currently, there's no standardized accepted methods to directly measure unbound intercellular drug concentrations. And although significant progress has been made in using modeling approaches to predict the effect of drug metabolizing enzymes in transporters and systemic exposure of drugs in preclinical species in humans, but we still have a long ways to go on this. So the next paper, I'm going to turn it over to Andrea to talk about the next one.
Thanks, Greg. So the sixth paper that we're going to discuss today is the ITC's recommendations for transporter kinetic parameter estimation and translational modeling of transport-mediated PK and DDIs in human. The first method that is discussed in this paper is the two-step method for uptake transporters. And in this method, a single low substrate concentration is evaluated at a single incubation time point. In cell systems, these incubations should be conducted under linear rate conditions at 4 and 37 degrees Celsius. Then the active uptake is determined by subtracting the passive uptake at 4 degrees Celsius from the total cellular uptake at 37 degrees Celsius. Transporter-mediated uptake can also be determined in the presence of inhibitors with this method. The limitations of this method is that there's limited membrane fluidity at 4 degrees Celsius. There's also a lack of specific inhibitors, and there is substrate-dependent inhibition, especially with the OATPs. In expression systems, active uptake can be determined by subtracting the uptake in control cells from the uptake in transfected cells. And the limitations of this method are that it assumes transport is an isolated process. It doesn't account for the bidirectional nature of passive diffusion, intracellular binding, metabolism, or active efflux. And data transformation is required for parameter estimation with this method. This method is most applicable for low permeable compounds, and this is the equation that they provide to use for the two-step method. To overcome some of the issues with the two-step model, a mechanistic compartmental model has been developed. In this model, plated and suspended hepatocytes are used, and this allows the model to account for active transport, passive diffusion, and intracellular and extracellular binding. Multiple substrate concentrations are evaluated in multiple incubation times at 37 degrees Celsius. The incubation time points are extended beyond the linear range to attain steady-state intracellular conditions, and this allows for a more accurate estimation of intracellular binding. However, it assumes that the intracellular binding is not saturated, and this may result in an overestimation of the fraction unbound in the cell. With this model, active efflux is not considered, nor is the internalization of efflux transporters. Metabolism is not incorporated in this model, and so uptake data obtained in the presence of ABT should be used for CYP substrates. This model is most applicable for compounds that are not metabolized or are only metabolized by CYP enzymes. And the paper provides these two calculations to use with this model. The mechanistic uptake model can be extended to account for metabolism. This is also conducted with plated and suspended hepatocytes and extended incubation times to attain steady state. But in this model, both the drug and the metabolites are measured. Data analysis for this model is more complex but has less error. It provides a mechanistic description of in vitro processes and can account for canicular efflux using sandwich cultured hepatocytes. These are evaluated using a single low substrate concentration for multiple time points. This model is most applicable for compounds that are metabolized by enzymes with no selective inhibitors, such as glucuronidation. And the ITC recommends determining Km and Vmax over clearance for in vitro extrapolation. And this is the equation that is provided for the mechanistic compartmental model with metabolism. E-Flux transporters can be evaluated in vesicles, as Greg mentioned earlier. Vesicles are best used with low permeable compounds. They're commonly used for polar substrates that are transported by the MRPs, MATES, and BCRP. They're also used for the inhibition of BSEP. They're not the test system of choice for PGP and BCRP since these substrates are lipophilic and have higher permeability, but there are some instances where they can be used for PGP and BCRP. 3 different compartmental approaches have emerged to allow for the determination of kinetic parameters for E-Flux transporters in monolayers. The first model is the 3-compartment model, which has a basolateral compartment, a cell compartment, and an apical compartment. And then an E-Flux ratio is determined in each one of these compartments. The E-Flux ratios are then used to calculate Km and Vmax and IC50. And here are some examples of those calculations below. Whoops, sorry. This method assumes that there's minimal lag in flux and similar passive permeability across the apical and basolateral membranes. And if these assumptions are not true, then this model is not valid. As the result of observed lag times in the monolayer flux of certain molecules, a 5-compartment model has been developed. This model includes a basolateral membrane and an apical membrane, and these membrane compartments serve as storage compartments when cellular concentration doesn't quickly reach equilibrium, which results in a lag flux time. With this model, the membrane to water partition coefficient is determined, and it's called Kp, and it's estimated in human liver microsomes. And then the Km and the IC50 is estimated in terms of the unbound drug concentration in membrane and cytosol, respectively. For both the 3 and the 5-compartment models, they predict similar intracellular concentrations following apical addition of substrate. But they differ in the prediction of intracellular concentrations for flux in the beta-A direction. This model is most appropriate when there's lag in flux time and for the prediction of tissues with basolateral exposure to drugs, such as the liver or kidney. And metabolism can be also incorporated into this 5-compartment model. The third model is the structural model and is the most complex model of the 3. It considers binding of the substrate from within the inner leaflet of the apical membrane with on and off rate constants and an E-flux rate constant from PGP into the apical chamber. It also estimates PGP E-flux active surface density. And then these parameters are used to calculate Km and Vmax with these equations shown here. This model requires the use of multiple initial drug concentrations and time points until steady state is reached between PGP-mediated E-flux into the apical chamber and passive permeability from the apical chamber back into the cytosol. Ki can also be calculated from the IC50 with this model, and this model accounts for basolateral transport. Some things to consider for in vitro estimation of uptake and E-flux parameters is that the probe substrate concentration should be well below the Km for the IC50 to equal the Ki. The ITC has not agreed upon a method for Ki determinations in polarized cell lines, so IC50 is preferred in this test system. Substrates requiring both basolateral uptake and apical efflux, such as digoxin, should use net values for inhibition to determine IC50. There's also been the evidence of substrate-dependent inhibition, such as with the OATPs, which suggests that transporters have multiple binding sites. So the ITC recommends determining inhibition with relevant co-medications as well as prototypical substrates. Pre-incubation may increase inhibition, and so incubation should be conducted with and without pre-incubation, and then incubation should be conducted at relevant physiological pH. With the exception of the oats and the mates, all major transporters can be evaluated in knockout animals. PGP and BCRP data from knockout models can be used to predict rate and extent of absorption. And if absorption, if there's complete absorption or minimal in vivo efflux, then the ITC says that you may not need to conduct a clinical DDI study of intestinal efflux. Knockout animal models can also be used for blood-brain barrier predictions, which will be discussed in the last paper. And the fraction excreted method is the preferred method, and it uses these two equations here for substrate and inhibition. And then this figure here shows that there's a nonlinear increase in the exposure as inhibitor concentration increases. Some things to remember with knockout animal models is that you should confirm that the compound exhibits similar pharmacokinetics between humans and knockout species. There have been species differences that have been noted, and so this should be assessed in vitro to support translation to human. There's also been the potential for up or down regulation of transporters and drug metabolizing enzymes in these knockout animal models, and so this may complicate data interpretation. Two models, static and PBPK models, have been developed for in vitro and vivo extrapolation. The static models are based on clearance concepts, and these models assume that there's a single transporter that dominates victim drug absorption, distribution, and clearance. This model also assumes that there's a constant concentration of inhibitor or substrate during dosing, and although this model may eliminate false negatives by assessing the worst case scenario, it can result in false positives. So to overcome some of these limitations, the PBPK model was developed. This incorporates permeability-limited tissue compartments to account for diffusional barriers, and an example is shown here below, which is the permeability-limited liver model. The PBPK models can be optimized with existing clinical data for improved predictability, and this is called a chop-down approach. The clinical data can bridge the gap between in transporter in vitro and vivo extrapolation by generating empirical scaling factors, but it should be noted that multiple scaling factors are going to have to be determined, and so a lot of the data is going to be required. And some of these scaling factors include inter-individual variability, cellular system differences, differences in transporter expression, differences in test system versus tissue activity, and allelic variance. So in summary, in vitro and vivo extrapolation requires information from numerous processes and organs. Each has its advantages and limitations in translating data to the clinic. Compartmental modeling is the best approach. Full kinetic characterization of transporters is required. Knockout animals can provide valuable information and can improve the understanding of human PK, but should be used with caution. Expression and localization of transporters is needed, and more work is required to determine scaling factors. And the last paper that we're going to discuss today is why clinical modulation of E-Flux transporter at the human blood-brain barrier is unlikely. So based on preclinical studies, it was thought that there was a potential for large DDIs that could occur due to E-Flux transporter inhibition. And this publication provides evidence for why these DDIs are not likely at the blood-brain barrier in humans. The blood-brain barrier is the tightest endothelium in the human body. As compounds increase in lipophilicity, they also increase in passive permeability. And so E-Flux transporters limit the CNS penetration of these drugs. Transporter function at the blood cerebrospinal fluid barrier can function differently than at the blood-brain barrier, and that's shown in this figure here. So here, this is the blood cerebrospinal fluid barrier, and PGP and BCRP function is that it secretes substrates into the CSF. But at the blood-brain barrier, PGP and BCRP efflux into the blood. PGP and BCRP are the two most studied transporters in the blood-brain barrier, and there have been some studies showing that inhibition of both PGP and BCRP has a synergistic effect. And this is shown in this figure here for lapatinib. So studies with knockout mice were conducted with lapatinib, and in BCRP knockout mice, The fraction excreted was 0.23, which resulted in a 1.3-fold increase in CNS distribution. And in PGP knockout mice, there was a 3.8-fold increase in CNS distribution. So when you add the fraction excreted of the BCRP and PGP, you get a 0.93 fraction excreted, which equates to a 33-fold increase in CNS distribution. And the actual result resulted in a 26-fold increase. So it should be noted that if you were to add just the fold increases here, you would get approximately a 5-fold increase if BCRP and PGP were both knocked out, but that would grossly underpredict the fold increase. So it's a function of FE, not a function of the fold increase. That should be taken into consideration using this equation here. However, when this was translated to the clinic, the same observation was not observed, and that's what's shown here in this figure. So again, the PGP and BCRP knockout mice results are shown here with elevated CNS distribution as you increase in concentration. But in the clinic, the highest concentration of inhibitor that's attained is approximately equal to the Ki, and so that would only equate to a 2-fold increase in CNS distribution clinically. And this was shown with cyclosporin and efavirenz. Cyclosporin is a PGP inhibitor, and the Cmax over Ki for cyclosporin is 1.6. And efavirenz is a BCRP inhibitor, and its Cmax over Ki is 0.06. So when these two compounds were co-administered with lapatinib, the largest DDI that was observed was a 1.9-fold increase. And so it should be noted that all currently marketed PGP inhibitors achieve unbound systemic concentrations that are approximately equal to the Ki or less. And so at most, you're going to impair blood-brain barrier efflux by 50%, which will elicit a 2-fold or less increase in CNS distribution. Other transporters that are present at the blood-brain barrier are MRP4. This is present in both human and rodent. It limits the penetration of 2 different compounds, adefovir and topotecan. However, in MRP4 knockout mice, there was less than a 2-fold increase in the exposure of these compounds. And so this just shows that there's a low risk of DDI even if MRP4 is completely inhibited at the blood-brain barrier. Numerous uptake transporters are also present in the blood-brain barrier. However, competitive inhibition is not likely due to the fact that the Km of these substrates is much greater than the Cmax that's attained at the blood-brain barrier. And furthermore, no clinical interactions have been noted for these substrates. So CNS exposure is defined by Kp, which is the brain-to-plasma ratio. And for a systemic DDI, the drug concentration is going to increase systemically, and that's going to increase brain exposure. But that does not necessarily mean that brain distribution is altered. And so a systemic DDI does not equal a blood-brain barrier DDI. And in fact, blood-brain barrier DDIs have not yet been recorded for any of the currently marketed PGP inhibitors. Some models that can be used to measure blood-brain barrier DDIs are brain capillary endothelial cells, non-cerebral cell lines, animal models, PET imaging, CSF concentration, and CNS pharmacodynamic effects. So in summary, DDIs were predicted based on knockout animal studies that showed complete loss of transporter function. However, complete inhibition of transporters at the blood-brain barrier in human is not likely. The unbound Cmax of compounds that are PGP inhibitors has been less than or equal to the Ki, which results in a PGP and BCRP inhibition of less than or equal to 50%, which would result in a 2-fold increase in exposure at most. In addition to this, no clinical induction of blood-brain barrier PGP has been reported, and there's been no clinical correlation between PGP function and CNS disorders. And with that, we've concluded all of the review of all the papers, and I'll pass it over to Matt.
Thanks, Andrea. Quick reminder, if you have any questions, please post those in the questions pane. We're running low on time, but we will get to just a few of those. And before we move into that, I wanted to share with you all a few upcoming events where XenoTech will be participating. On September 17th, we will be at the IQPC Clinically Relevant Drug Transport meeting in London. Our European account manager, Tom Zaleski, will be there. Please feel free to meet with him and discuss drug transport or anything else that you're interested in talking about. Also, we will have a booth at the 10th International ISSX meeting in Toronto, Canada on September 30th. Our booth number is 301, so I invite you to swing by the booth, introduce yourself, and talk with our representatives on site. With that, let's run in just maybe time for about three questions, Greg, and your team, and then let's close this up.
Thanks, Matt. So the first question we have is about the expression of MRP3 in 4 in cholestasis. I know if MRP2 is downregulated, MRP3 and 4 will increase in expression. I don't know if direct inhibition of BSEP will cause increase in the expression of MRP3 and MRP4. The next one.
Sure. There's a question about right now bilirubin and ALT and AST are what are monitored in the clinic, but there's no direct link to BSEP. And the question is, would it be better to investigate bile acids? I think bilirubin and ALT right now are kind of a standard of overall liver toxicity, and you're correct, it's not a direct link to BSEP malfunction. I think right now in preclinical studies, or at least in animal studies, they do measure bile salts or bile acids in the serum of animals, but I'm not sure if that is done in humans.
Now, there's a couple other questions. We're going to respond to some people directly. Some questions about lysosomal trapping. We do have a paper and a link to the paper on our website about lysosomal trapping. So that's one place to find that information. So with that, we're going to end the question and answer session for now, and I'll turn it back to you.
All right, thanks, guys. We're going to go ahead and wrap up today's session. We're running a little late here, but thank you all for taking time out of your day to join us, and thank you to the Drug Transport Group for their presentation. If you do have any extra questions or concerns, please contact us directly at info@xenotechllc.com. We will follow up with you immediately. So thanks again for joining us today, and have a great rest of your day.